So today we're going to start talking about Protist. This is chapter 25 of your book. This will be the first kingdom of the domain Eukarya that we're going to discuss. So the Protists are pretty much just a catch-all for anything that is not animal, plant, or fungi. So that's why it is a very diverse group of unicellular eukaryotes. Here we actually see two protists. One is consuming the other. And a lot of the protists are able to change shapes. So our first eukaryotics, as we've talked about in previous chapters, were thought to be evolved about 1.8 billion years ago. And they were thought to come about due to endosymbiosis. And so here are several examples over the last 1.8 billion years and how they became even more complex from their origin of replication. And so they are very diverse. Our early eukaryotes had a nucleus, so they had that membrane-bound nucleus. They had a plasma membrane, they had a cytoskeleton, and they came in a variety of shapes. And then around 1.2 billion years ago, we have our first rig algae. They are the oldest eukaryotic fossils that have been found and resolved taxonomically. Other fossils from this period include our green algae, amoebas, and other multicellular protists. And then more recently, about 500 million years ago, our protists became larger. So they are large multicellular eukaryotes. They have sexual life cycles instead of just an asexual life cycle, and many go back and forth. And they develop photosynthesis. And so during this time, during the Cambrian explosion, is when we had a large increase in our size and diversity of the eukaryotes. So what supports that endosymbiotic theory is that there are lots of similarities between eukaryotic cells and our predecessors, the archaea and the bacteria. So the eukaryotic DNA replication enzymes, archaeal in source, transcription, translation, archaeal, cell division apparatus, archaeal, so that mitotic spindle that we've talked about, endoplasmic reticulum, both archaeal and bacterial, mitochondrion from bacteria, and our metabolic genes such as our chloroplast, and our chloroplast also most recently from bacterial. And if you remember the evidence of endosymbiosis, that you have the double membrane on the chloroplast and the mitochondria, so you end up with that double chloroplast because when they were taken in via endocytosis, they kept the membrane of the cell that took them in, the host cell, and the cell wall that they had. And then the chloroplasts were thought to come after, and that's why all eukaryotic cells have mitochondria. A lot of people say, well, plants have chloroplast and animals have mitochondria. But no, plants, animals, everybody has mitochondria. And so that's why it's thought to be the first that formed via this endosymbiotic relationship, and then the chloroplast came next. And then we also had lots of other membranous type things that developed via the endosymbiosis. And so this is what we call serial endosymbiosis, where we had multiple rounds of endosymbiotic to form our eventual mature eukaryotic cell. And so a lot of the evidence that supports this, those two inner membranes, and a lot of times in this book especially, you're going to see the chloroplast called plastids. So when you see plastids, it's the same thing as our chloroplast. The replication of the mitochondria and the plastids are similar. They have their own circular DNA, very similar to bacterial DNA. They transcribe and translate their own DNA into proteins within the organelle itself. Ribosomes of the mitochondria and the plastids more similar to bacterial than to the eukaryotic ribosomes. DNA sequence says that the mitochondria arose from a single phyla of photobacterium and that the plastids came from a single common cyanobacterium ancestor. So lots and lots of evidence shows that the endosymbiosis is how we evolved from the prokaryotes to the eukaryotes. All right, so let's watch a couple videos.
Life on Earth emerged at least 3.5 billion years ago as prokaryotes. These are the simple unicellular organisms. They have a membrane on the outside and a wash of cellular machinery inside, all mixed together and touching and sharing the same environment. Now, this worked, certainly. Life chugged along this way for nearly half of the history of life on Earth. But then, 1.8 billion years ago, something remarkable happened. Something that led to a tremendous shift in the scope and complexity of life. Something we should all be grateful for, because without that leap, we would not exist. Cells started to contain cells. Now, this isn't generally how it's talked about in science class. There you hear that eukaryotes have, quote, membrane-bound organelles. These are areas of the cell that are separated from the rest of the cytoplasm by membranes, just as the cell itself is separated from the rest of the universe by its membrane. It turns out, different activities require different conditions, and these cells within cells allow for those different conditions. That, in short, is the secret of eukaryotic success. But how did it happen? Well, over decades of study, we've determined something shockingly peculiar, something so odd that it makes us kind of mad that we now discuss it as if it isn't the miracle it is. 1.8 billion years ago, a cell consumed another cell, but then it didn't digest it. It let it reproduce inside of it, and they lived together and over time became the same organism. Or did they? This is what we call endosymbiotic theory. Mitochondrion appeared when the consumed cell was adapted to live in an oxygen-rich environment, and chloroplasts appeared when the swallowed cell was photosynthetic. This idea was deeply controversial when it was first proposed, but as data have continued to come in, endosymbiotic theory has been able to explain more and more about the realities we find. For example, that chloroplasts have their own DNA, which they use to create the proteins required for their function. And as we dive deeper into the microcosmos, it just becomes obvious that this happens. This is Paramecium bursaria, a single-celled protozoa that has several hundred algal cells from the genus Chlorella living in its own cytoplasm, making it green. The algae live inside Paramecium bursaria, providing it with fuel in the form of sugar and other substances produced via photosynthesis. And Paramecium bursaria provides protection for the algae from algae eaters and viruses. P. bursaria is regarded as a predatory protozoa. It feeds on bacteria, small organisms, and yes, algae. And because of that, it's often thought that the algae in it are temporary symbionts engulfed by Paramecium bursaria's feeding behavior. But in fact, many other protozoa acquire algae in that manner for temporary use, but that is not the case for P. bursaria, its symbionts are continuously inherited from generation to generation through cell division. The symbiotic chlorella guide the paramecium to well-lit areas so they can photosynthesize more efficiently, and the mutual relationship is extremely beneficial for the paramecium, even when chlorella-containing paramecium cells are put in a nutrition-free saline solution. They could survive for more than 3 months, while cells that didn't have chlorella died within a week. This is another single-celled organism with endosymbiotic algae. It's a testate amoeba, a kind of amoeba that builds itself a shell. This species, like some kind of sculptural artist, pulls bits and pieces of mineral from its environment to create these amazing-looking homes. You can see the amoeba extending from the opening of the shell, and you can see the green algae in its cytoplasm. Just like Paramecium bursaria, the algae use sunlight to produce food, sharing it with the amoeba, while the amoeba provides protection. Some unicellular organisms don't need oxygen for growth. Indeed, the presence of free oxygen can affect them negatively or even kill them. These organisms are known as anaerobes, such as this one, Metopus. It's an anaerobic ciliate we find in pond sediment, and it has an endosymbiotic relationship with methanogenic archaea. Now, we haven't talked much on this channel about archaeans, but they are the third domain of life, along with bacteria and eukaryotes. And like bacteria, they are prokaryotic. We can't wait to do our episode on them someday soon. Many of the single-celled eukaryotes living in anaerobic environments contain symbiotic prokaryotes. Some of these prokaryotes are methanogens, meaning they can use free hydrogen to generate energy and methane. The advantages of having these symbionts are not fully understood, but while Metopus can live without the symbionts, they grow faster when they have them. Endosymbiosis occurs in multicellular organisms as well. This is a freshwater relative of jellyfish and sea anemones, Hydra. It's simply stuffed full of algal symbionts. We collected this Hydra from a nearby pond and cultured it in our aquarium. The benefits provided by the symbiotic relationship here have been well documented, with scientists actually tracking how carbon moves from the environment into the algae and then into the Hydra. And studies have shown that up to 69% of the caloric requirements of the Hydra is satisfied by its algal symbionts. Nice. So we see some organisms temporarily pull in symbionts, others pass them from generation to generation. Some can survive without them, and some cannot. When we look at the algal cells in P. bursaria, we're forced to ask if those cells are part of the organism, or if they're simply cells of one species living in the cells of another. If that's the case, it's worth asking whether the mitochondria in you are you at all, or if they are just another extremely successful species of prokaryote that is particularly reliant on its host cell. As we look deeper and deeper down, the line between organisms is harder and harder to find, which is why, if you think hard enough, you might begin to feel like our cells are more than just ourselves. Thank you for coming on this journey with us as we explore the unseen world that surrounds.
What if you could absorb another organism and take on its abilities? Imagine you swallowed a small bird and suddenly gained the ability to fly. Or if you engulfed a cobra and were then able to spit poisonous venom from your teeth. Throughout the history of life, specifically during the evolution of complex eukaryotic cells, things like this happened all the time. One organism absorbed another, and they united to become a new organism with the combined abilities of both. We think that around 2 billion years ago, the only living organisms on Earth were prokaryotes, single-celled organisms lacking membrane-bound organelles. Let's look closely at just 3 of them. One was a big, simple, blob-like cell with the ability to absorb things by wrapping its cell membrane around them. Another was a bacterial cell that converted solar energy into sugar molecules through photosynthesis. A third used oxygen gas to break down materials like sugar and release its energy into a form useful for life activities. The blob cells would occasionally absorb the little photosynthetic bacteria. These bacteria then lived inside the blob and divided like they always had, but their existence became linked. If you stumbled upon this living arrangement, you might just think that the whole thing was one organism, that the green photosynthetic bacteria were just a part of the blob that performed one of its life functions, just like your heart is a part of you that performs the function of pumping your blood. This process of cells living together is called endosymbiosis, one organism living inside another. But the endosymbiosis didn't stop there. What would happen if the other bacteria moved in too? Now the cells of this species started becoming highly complex. They were big and full of intricate structures that we call chloroplasts and mitochondria. These structures worked together to harness sunlight, make sugar, and break down that sugar using the oxygen that right around this time started to appear in the Earth's atmosphere. Organisms absorbing other organisms was one way species adapted to the changing environmental conditions of their surroundings. This little story highlights what biologists call the endosymbiotic theory, the current best explanation of how complex cells evolved. There's a lot of evidence that supports this theory, but let's look at 3 main pieces. First, the chloroplasts and mitochondria in our cells multiply the very same way as those ancient bacteria, which are still around, by the way. In fact, if you destroy these structures in a cell, no new ones will appear. The cell can't make them. They can only make more of themselves. Second piece of evidence. Chloroplasts and mitochondria both contain their own DNA and ribosomes. Their DNA has a circular structure that is strikingly similar to the DNA of the ancient bacteria, and it also contains many similar genes. The ribosomes, or protein assembly machines of chloroplasts and mitochondria, also have the same structure as ribosomes of ancient bacteria, but are different from the ribosomes hanging around the rest of the eukaryotic cell. Lastly, think about the membranes involved in the engulfing process. Chloroplasts and mitochondria both have 2 membranes surrounding them, an inner and outer membrane. Their inner membrane contains some particular lipids and proteins that are not present in the outer membrane. Why is that significant? Because their outer membrane used to belong to the blob cell. When they were engulfed in the endosymbiosis process, they got wrapped up in that membrane and kept their own as their inner one. Surely enough, those same lipids and proteins are found on the membranes of the ancient bacteria. Biologists now use this theory to explain the origin of the vast variety of eukaryotic organisms. Take the green algae that grow on the walls of swimming pools. A larger eukaryotic cell with spinning tail structures, or flagella, at some point absorbed algae like these to form what we now call euglena. Euglena can perform photosynthesis, break down sugar using oxygen, and swim around pond water. And as the theory would predict, the chloroplasts in these euglena have 3 membranes, since they had 2 before being engulfed. The absorbing process of endosymbiotic theory allowed organisms to combine powerful abilities to become better adapted to life on Earth. The results were species capable of much more than when they were separate organisms. And this was an evolutionary leap that led to the microorganisms, plants, and animals we observe on the planet today.